Now, you know that organic chemistry is the study of compound that contains what? Carbon, whose atomic mass unit is 12.011 and atomic number to be 6. Its symbol is represented as C. Now, where 100% of the interaction or bonding is between carbons, you can have it either in single, double, or triple bonds. We have other bonding between carbon and other elements, like for example in percent wise using this pie chart, majority of the interaction with carbon is with hydrogen, which are usually single bonded, while another small fraction is between carbon and oxygen, some to be either single bond and some others could be double bond. Another bonding is between carbon and nitrogen, and this here usually is found in amino acids, and this is usually in just single bond, but sometimes can be seen in double bond or triple bond. And finally, the rest are all just derivatives, like either carbon with sulfur, carbon with any other metals that you might think of, and this takes up little fraction of this pie. Now we have a brief understanding of what organic chemistry is all about, given the molecular structures for these compounds. By analysis, we notice that they have a unique design that is common in all of them. What we see is that they are under the family of the amino acids, and these are known to be the building blocks for proteins. And since we notice that they are amino acids, structurally we see that they are identified based on this unique formation, which is common in all of these compounds highlighted in yellow. And what we see is that there is an amine group attached to a carbon, and while on the other side we have a carboxylic acid. The variations of amino acids is based on the carbon that is attached to the hydrogen, and this hydrogen changes as you move from this here to now this carbon chain attached to this right here, and this and what we have over here, and this one right over here. This variation shows the unique diversity of amino acids and how they function, where on one end, by naming based on the R group, this here represents glycine because it has only hydrogens attached to this carbon, while the next one here has one methyl group attached to that carbon, and this is called alanine. And then moving from this here to this structure right here, we have that methyl group now attached to a phenyl group, and this is a derivative of alanine, which is called a phenyl alanine. And next is moving on to this structure, which is a long carbon chain, and at the end it has a amine attached to the last carbon. This here is called lysine. This here is followed by without a nitrogen, but then the long carbon chain has a branch carbon, which is known as leucine. And then finally here is moving on to this long carbon chain that is the R group, but here we have a sulfur that is bonded to a methyl group, and this is known as methionine. Now, based on the changes in the R group as highlighted in orange, some amino acids, they share similar properties. Like for example, some of them are soluble in water, while some others are insoluble in water, usually is hydrophilic or hydrophobic respectively, and while some other structures tend to be more acidic, while some others are basic, which makes those type of compounds to be known to be called amphoteric. Now, this unique behavior tends to be less from the carbon chain that is on the R group, but more influenced by what? The unique functional groups that are around the R group, either in this part here which is sulfur, or we have this here, which is our amino group. And this here is really incredible to see how these are many more behaviors that you notice, like for example how phenylalanine is quite unique compared to another structure that is similar to this as shown right here, where phenylalanine over here is different from this structure right over here, which you notice has a nitrogen attached to the ring, and this here is known to be called a tryptophan. Now, based on this here, you might imagine that they might behave differently, but no, not at all, because they do have the same properties, which is they are non-polar, which means that they are not water-loving or hydrophobic. However, compare phenylalanine to another amino group with similar structure but with an additional functional group, which in this case is shown right here, where this amino group is known to have a hydroxyl group that is attached to the phenyl group. This amino acid is known as tyrosine, and based on the fact that the hydroxyl group is attached to the phenyl group, this hydroxyl group makes this amino acid to be polar, or in this case known to be hydrophilic. Or from phenylalanine, we change the R group that has the phenyl there to another ring that has a different structure as shown right over here. And this structure here shows that the R group has two nitrogen atoms that are in a 5-membered ring, and this here is known to be called histidine. In addition, this amino acid is known to be an essential amino acid because our bodies, humans specifically, can't make these amino acid, but rather we have to obtain this from our diet. In addition, also, these distinct features between phenylalanine and that of histidine because of the nitrogens here implies that the lone pairs on this nitrogen here will be essential for what? The picking up of protons from water in order for these to behave more like a base. And this here is characteristics of histidine compared to phenylalanine, and how phenylalanine tends to be similar, of course, to tryptophan, despite the fact that we have a nitrogen present, and while phenylalanine is really different from that of tyrosine because of hydroxyl attached to the ring. Now, analyzing these three amino acids, they are known to be what? A non-essential amino acids, which means that your body can make these amino acids. Now, looking at this first structure here, this has a R group, which is in this case represented as a methyl attached to the amino acid functional group, and so this here is known to be called alanine. Now, alanine is known to be what? Really non-polar, which means that it is not water loving, or it is hydrophobic because of the methyl group that is present as the R group. Now, comparing alanine to the next amino acid, which is what we have over here, this amino acid here is called aspartic acid, because we have a carboxylic acid group present at the R group end, and what that implies is that once water comes into play, water is going to interact in such a way that the oxygen on water is going to pick up this proton, and this will result in the formation of what? A negative charge that will be present around this carboxylic acid region right over here. Now, from aspartic acid to this particular structure, we have the methyl group now attached to an amide. This here is called asparagine, and the presence of this amide here, which is making this asparagine to be amidic, is known to affect it in a way that it is known to be what? Polar. And this means that it is water loving compared to alanine, which doesn't have the amide, and this makes alanine to not be polar. Now, after observing all the amino acids or majority of them on this video, we observe some interesting things about the R groups as shown right over here. And what we see is that the functional groups that are noted here are supported by a hydrocarbon framework that looks like either a linear system or a chain, or we have a branched chain that looks like this, or a ring that looks like this. Based on this video, you observe my drawings that I performed, and the question that will be addressed on the next video is why use different drawing methods and why is it more effective for one versus the other in organic chemistry. I hope to see you all on the next one. Stay smart as always and believe in yourselves.